Our next speaker is Becky Thompson from Leeds, and she's going to tell us about cryo-electron microscopy at Leeds.
Hi, yep, I'm on. Oh, that's very loud. Hi, my name is Becky. I'm from the University of Leeds, and hopefully I'm going to tell you a little bit today about some of the journey that we've been on as a facility in the last year or so. So for those of you who don't know, the University of Leeds invested a total of £17.5 million. That was 16 million from the University of Leeds and £1.5 million from the Wellcome Trust in structural biology at Leeds. So this was a huge investment in both electron microscopy and NMR. So as part of this investment, we were fortunate enough to purchase 2 Titan Cryos microscopes. You can see the time-lapse Cryos 2 being installed in the background as we speak. We had Cryos 1 installed with a Falcon 3 direct electron detector, and Cryos 2 with a K2 at the end of an energy filter, and also with a phase plate, so with all the toys. Yes, I love this time-lapse. You can see the field emission guns just about to go on at the top, up there at the back. So both microscopes were installed into the same space. The space is actually fascinating, a disused television studio, which was a bit of a university dumping ground before we took it over. We got the funding announcement in June of 2015. We placed the order for the microscopes, and building work had begun by the December of that year, and we had the building handed over and the installation of both microscopes well underway by September 2016, with us starting to collect some very high quality data by Christmas of that year. So it felt like a very long journey, but I think chatting to other people, we were probably pretty good on our timescales on that one. As Alan at the end there, just like trying to, what's up with the heck? So yeah, Alan was one of the main engineers for us who was the lead engineer on this particular install. So I'd just like to share with you a couple of lessons that we learned while we were setting up this facility. And the first one was really that environment was all important. So we were immensely privileged to be able to design and refurbish a space that really, really met our needs. And a big part of that for us is having dedicated sample preparation space directly next to our low humidity, so less than 20% relative humidity freezing room. The low humidity, if anyone, I know that a number of people here are going to be getting new Titan Cryoses in the next couple of months and years, and this is super exciting. And if you are fortunate enough to be designing yourself a new facility, I would say that a low humidity freezing space is amazing. We moved into it, and the quality of our grids increased, transfer ice went right down, everything got a lot cleaner and a lot nicer, which was fantastic. And then I kind of got used to it, and then it broke the other week, and I remembered just how much I love the low humidity. That's a real bonus for the facility. Because of the way we've set the facility up, we're right next door to our dedicated tissue culture facility. This enables us very easily to work with biosafety level 2 specimens, and also to be developing our cell work, which is great. So, as I said, where the Cryoses are is a disused television studio. So obviously on the left here we've got our Titan Cryoses, both of them in the same space. It's not normally that tidy, put everything in the cupboards for that photo. From a user's perspective, you literally enter the room in order to load the specimens into the microscope. On the right you can see we've got the workstations where the users control the microscopes up in a viewing gallery, or the director's deck, which overlooks the microscope room, which is a really fantastic setup because it means that as a user you can go into a nice office environment with your cup of tea and set up your data collection rather than having to sit in the room with the microscope, possibly in a lab coat. So I've just said that we've had the luxury of being able to design a facility from scratch, and we really have, but of course we've had to work within the confines of our existing building. So for those of you who've never visited the beautiful campus of Leeds, here's our wonderful brutalist architecture here. It's actually a grade 2-star listed building, would you believe? So as part of our refurbishment, we had to keep some of the original features, including this rather lovely recording sign on the right. The architects were really, well, the people who deal with the grade 2-star listing status were extremely complimentary about our lovely skateboard ramps here, which skateboarders just love, and they wouldn't let us get rid of them. Nightmare. Anyway. So the great thing about us refurbishing a space was that we were able to move pretty quickly. Like I say, from the announcement of the funding to us getting the building handed over, it was significantly less than a year. So that was a real bonus for us. Of course, if you have to build a new space, then it can take a lot longer than that. So lesson number 2, getting a Cryos is only the beginning. We didn't really anticipate the increase in admin and all of the boring stuff that was going to happen and come on board with that. And I would say to anybody who's going to be getting either a feeder microscope or a new Titan Cryo microscope, don't underestimate how much extra admin and things that you need to do. For us, the really critical bit was starting to try, and we're still not there yet, getting good systems and processes in place to keep communication up within the facility, keep everybody on board with what's happening, and to streamline the booking process. I'll talk a little bit more about the booking process that we've adopted a little bit later on. I know that a big theme here, and that is something that I've chatted to a number of you with today, has been training. So of course it's fantastic that the field is expanding in the way that it is, and that we're getting these huge numbers of new microscopes, but how do we deal with training all of these new users? So just briefly in terms of what we do at Leeds, is we tend to start any of our users off who want to do electron microscopy on a starter microscope. So for us that's either a JL 1400 or a T12, move them up to learn cryo on the F20, and then move on to learning on the Titan Cryos. Now of course this is a huge time investment, and for some people it just doesn't work in the context of their project. So again, we're still learning kind of the best ways to set up our training programs to mean that when a user hits the Cryos, that not literally, when a user gets on the Cryos, they are able to fully understand and make sensible decisions about the kind of data they want to collect, but yet maybe don't have to spend 3 years of their life, like I did, learning, or for more, learning cryo right from scratch. So I'd be very interested to chat to anybody who's got some ideas about this. Talking about those feeder microscopes, for us, we've obviously got 2 Titan Cryoses. This is an amazing resource. One of the things that's becoming apparent to us is how do we feed this pipeline? So how do we keep up the level of activity inside, in each of the labs? How do we increase the number of research groups coming to use the microscopes and get their projects ready to go into the Cryos and to generate high-resolution structures? And for us, having the on-site availability of 3 of these microscopes, which enable users to assess samples both in negative stain and in cryo, is a massive advantage for us. And I know that, again, that's something I've spoken to a number of you today about. Access to these screening microscopes is possibly another bottleneck to getting more projects into Titan Cryoses. And finally, the big question, data storage and processing. So these machines are just data monsters. They create so much data. How do we store this? How do we store this long term? And how do we process our data? So I just want to provide you guys with a little bit of information about the setup that we have. And of course, I'm very happy to share more details on this if anybody is looking to develop this kind of thing in future. So in terms of getting the data off the microscope, so the Falcon 3 rather nicely comes with a lovely offload server where the data directly writes to this very large 60 terabyte server. This is then linked by 10 gigabit uncontested Ethernet to our main storage servers, and therefore we can just kind of pull data off at our leisure. We're going to be implementing a similar system on our Cryos 2 as well with the K2. So getting the data off the microscope, not so much of an issue at the moment, but then storage. So when we first got this investment, we created 2.6 petabytes of raw storage. We filled up, I think we worked out was 0.9 of a petabyte, which is quite terrifying. So that includes raw data and the processed data, but we are really generating data at an astonishing rate, and I think that long-term data storage is going to be a massive question for us. At the moment, the kind of plan is to have a multi-petabyte tape library, but we really don't want to do that. So we're kind of looking into other alternatives for long-term storage. And of course, the interesting bit, the processing. And of course, it was a fantastic talk this morning from Eric talking about the revolution in GPU accelerated processing, which we've taken full advantage of. So we have a number of different GPU workstations, which we use to do the majority of our processing on. I think, I don't have the numbers, but I know for a fact now that our CPU clusters are now not particularly utilised fully. And I think this was a trend that will continue, and we're going to be investing further in GPU-based workstations. Cool, so the third and final lesson that I'd like to share with you all is don't buy even-numbered Cryoses. And I know this is something that Alistair said, and yes, a number of people have noticed in the field. So so we were very fortunate to purchase 2, and it's a good job that we did, because Cryos 2 has been, microscopes like children. You're not supposed to have favourites. And I don't. I love them both equally. But Cryos 1 is like the child that does their homework and is very well behaved and never gets into trouble and produces lovely data. And cryo number 2 is like the one that needs a lot of hand-holding and persuading, but when it actually produces data as a microscope, it's fantastic. The on-grid contamination rate is very low, the stage is very stable. We've just had a number of communication issues between the K2 detector manufactured by Gatan and the FEI microscope. We're hoping that we're very, 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 very close, days, hopefully, to nailing the final set of problems that have plagued this microscope, and then we'll be away. Because it's been very frustrating for us. Cryo-2 is really our microscope of choice for any tomography projects, and of course it's got the phase plate, which I'm desperate to play on. So yeah, we're really hoping that we'll be getting that up and running very soon. So we officially opened in January of this year, and we had Jeremy Farrer of the Wellcome Trust coming and cutting the ribbon, and we can see here we have our Vice-Chancellor, Alan Langlands, Sheena Radford, who's the Director of the Asbury Centre and was instrumental in us getting the funding from the university. For those of you who probably know Neil Ranson, who's the Director of our EM facility, looking very happy, and of course Jeremy Farrer, Director of the Wellcome Trust there. So that was a good day, and for those of you who haven't seen it, there's a very good cake associated with the kind of opening on Twitter. There's kind of some small giggles from the people who have seen it on Twitter. It's a big cryo cake. It's not that exciting. Fab. So I've told you a little bit about the facility. I'd now like to just show you some examples of some of the really exciting science that we've been doing at Leeds in the last, basically, I think almost everything I'm going to show you is since January. So I'm going to start off with some examples based around structure-based drug design. I know that a number of you in the audience are interested in looking at inhibitor binding and things along these trends. So the first example is work done by Rachel Johnson, who's in Steve Munch's lab, and she was investigating a membrane protein called BC1, which is a component of the electron transport chain. And she solved a number of these structures now, including the apo form of this protein and an inhibitor-bound structure. And both of these, well, all of the structures she solved now, I think it's 4 in total, are between 4 and 4.5 angstroms. So in this case, we can see in the inhibitor-bound form, she sees strong density for the inhibitor, which is not present in the apo form. So this is, so like I say, I think she solved 4 of these since Christmas. If you have a look at the maps, they were both collected on different microscopes, different data collection parameters. There's 4 times more data in the inhibitor structure, but not a huge amount of difference in total resolution. So this is an interesting test for us, and we think we can probably be pushing the resolution a little bit further. So we'll be looking to do that in the coming months. A second example is also an example from Steve Munch's group, this time from Sean Rawson. So for those of you who were here last year, you would have heard Sean talk about a protein called IGPD, which he's using as a model protein to look at a number of different things. So Sean initially did the structure of the Arabidopsis form of this protein with the inhibitor bound. There is also a yeast form of this protein which binds the same inhibitor, but in yeast the inhibitor is 10-fold more potent. So Sean was interested to see if he could use electron microscopy to rationalise why there was the difference in this change in potency. So as you can see here, in zoomed-in views of the map, we can see the inhibitor binding in both maps. And we can see that the global fold of the protein is very, very similar between both the Arabidopsis and yeast form. And if we have even a look at the inhibitor binding site, it's very, very similar between both the yeast and Arabidopsis form of this protein. So but using these structures, Sean was able to elucidate the fact that this extra 15 amino acid insertion loop inside the yeast form, even though this loop is quite distal to the binding site, this was modulating the binding site for this inhibitor and basically changing the mechanism with which this was acting, just explaining the change in potency. So this is a really nice example of how we can not just visualize inhibitor binding, but also start to maybe rationalize some of the reasons for its action. So another theme that we've seemed to have developed in Leeds at the moment is receptor binding, and we've done a number of different projects now looking at different receptors, how they bind, so I'd like to share a few of those with you. So the first one I'd like to share with you is BK polyomavirus. So this is a class 2 human pathogen. It's a leading cause of kidney failure and kidney transplant patients. And this work was done by Dan Herdis in Neil Ranson's lab. So as you can see, we're zooming into this 3.8 angstrom map now, and it's going to be showing the asymmetric unit of this protein, of this virus. So we can see here we have, this is the asymmetric unit, and each of these coloured proteins or monos is the VP1 protein coat unit. So for this virus, I should say, Dan has solved, I don't know now, about 5 or 6 structures of BK at ever increasingly iteratively better resolution as we've been kind of going along. He's absolutely sick to death of doing it, so I think that this is the one that he's going to stick with now, and he's going to start doing BK in more interesting scenarios. Yeah, because he kept on getting unlucky with the microscopes not quite behaving, and with it not quite behaving as it should. So he was quite pleased to get this high resolution structure in the end that he's been able to build into. What Dan was interested in doing in this case was looking at a receptor. So there's a particular receptor which is proposed to be a major mechanism of cell entry. So I'm just going to describe a little bit about the methods here because it was very interesting. So Dan has made the virus, and the virus is at reasonably low concentration. And Dan developed a protocol by which he was performing multiple applications to a grid. So he took his grid, which had a thin carbon film across the whole surface, added on some of his virus solution, blotted away, added some more on, blotted away, added some more on, and in that way was able to build up the virus concentration on the grid. And in this particular case, he was then able to, in the final step, add an oligosaccharide, which was part of this important receptor, onto the grid. And then lo and behold, when he solved the structure, rather pleasingly, he saw these lovely little oligosaccharide receptors binding 5 on each penton. So this was a really nice example of how we can use EM to look at a relatively small non-protein receptor. So in the second example I'd like to show you is probably Leeds' favourite virus, cowpea mosaic virus. So we've done a number of different structures on this over the years, including empty forms and with different genome components inside of it. And a group at Leeds is interested in developing what's known as affimers, or non-antibody binding proteins, and then potentially then using these in the field to develop tests for plant pathogens in the field. So this was really kind of a test case and a bit of a proof of principle. And also before Emma Hesketh, who's in Neil's group, had done this work, nobody had actually directly visualised by electron microscopy any of these affimers binding to any target. So Emma actually collected this data at EBIC, which was fantastic. And on the left, you can see the sharpened form on the map, form of the map, and we have in green and blue the capsid, and then in this light purple shade, we can see the affimer is binding on the two-fold axis. So an affimer is about a 12 kilodalton receptor, and it consists of this central alpha helix and then some beta sheets. You can see we've got in the unsharpened map on the right, we have really quite nice density for the helix, although the density for the beta sheets is being slightly washed out because it's on the two-fold axis. It's essentially being averaged out across the two different axes. It also wins the award for the rudest structure to come out of Leeds so far. And the final example in this category I'd like to share with you is, again, work by Dan Hurdis in Neil Ranson's group and collaborators. And I know a number of you in the audience work on picornaviruses, which is why I wanted to throw this one in here. Dan solved another structure of a class 2 human pathogen, this picornavirus, with, again, about a 12 kilodalton proteinaceous receptor. This receptor actually has 2 domains, but we're only visualising the first domain here, presumably because the second domain is flexible with respect to the first. So that, again, is a really nice example of the kind of projects that we're doing, and Dan's going to be looking to now have a look at doing this structure again with some possible inhibitors in place. So, my favourite bit of structural biology, looking at things that no one's ever done before. So, probably one of the most exciting things that we've been doing is looking at, like I say, biological specimens where nobody's developed or looked at high-resolution information of these things before or built models. And probably, again, you're not supposed to have favourites, but probably my favourite structure to come out of Leeds so far is this one that Emma Hesketh did in Neil Ranson's lab. And this is of a Gemini virus. So this is 2 T=1 icosahedrons, which have basically been together, and nobody knows exactly how that works. So it has the so this particular virus was solved. Well, the map was generated at about 3.4 angstroms. It has the native genome inside, so it has a single-stranded circular DNA genome inside. And as you can see, the density is quite beautiful. And was able to use COOT and RefMac to first of all build and then refine the structure. So in the colours here, we can see that this is, so I should say this structure was generated with C5 symmetry, and we have an asymmetric unit seen here. And this is data collected on cryo-tomography. So the protein, just being able to visualize this, the protein capsid was immensely exciting. But here we have the unsharpened map, and we can see in grey, this is where Emma built in the protein density. So she built in the protein and went, there's some extra stuff. And it turned out the extra stuff basically corresponds to bases and nucleotides, which are going to be coming into view just now. So this was immensely exciting, and Emma's working at the moment on developing, basically trying to rationalise now how this immensely peculiar biological structure is put together, because she thinks that the genome is having a really key role in this. So watch the space on that one. And the final example in this category is we have done generated a map about 3.5 angstroms of a luteovirus, and this will be the first structure of any luteovirus solved to sort of high resolution. So this was again a map done by Emma in collaboration with Mim Walden, who's in George Lomonosov's group in the JIC in Norwich. I should say they also generated the previous sample as well. And they basically express these proteins in plants, these virus-like particles in plants, which is really fantastic. So you can see here we're at a reasonably good resolution, and as a result we were able to start obviously doing de novo model building. So this is a bit of a shout out to CCP-EM, because I was very fortunate to come on the CCP-EM model building workshop a couple of weeks ago now, which was fantastic. Had a really great time, and it was brilliant to be able to interact with some of the tutors and actually make quite a huge amount of progress in terms of generating structure in just the very short workshop. So if any of you are interested in doing any of this kind of stuff and there is a CCP-EM workshop for it, then I can highly recommend going, because they're always great. So for me, the first stage in building this model was to put it through Buccaneer, and so I should say I first segmented the map. So we took the huge great big virus. It's a T=3 virus. So first of all, segmented out the T=3 subunit, then take out one monomer, and then try and build into the density of that one monomer. So Buccaneer has done a job here. It's done something which is good. So this bit is probably fairly sensible. These bits are just a bit random, and there were some bits where the sheets were in backwards and that kind of thing. So we then took it into Coot, where I've managed to clean it up and build in basically complete the work that the Buccaneer started. And we're going to be looking to put that into RefMac and Phoenix and refine it in the weeks to come, which is very exciting. So I've shown you some of the really exciting maps and models which have come out of Leeds in just the last couple of months, and hopefully this is a kind of a trend that we're going to continue going on into the future. But I'm conscious that a lot of you are going to be, so I'm conscious it's a very mixed audience. Some of you guys are real cryo-EM experts, and that there's a lot of people who are reasonably new to the field and are maybe thinking about how to develop a pipeline, and okay, right, I've got my protein and I know I want to do a structure by cryo-EM, but where do I start? So I just want to give you a flavour of the kinds of things basically the kind of pipelines that all of the structures that you just saw went through. So for us, the first step is always biochemistry. So again, nailing that. So I think the phrase garbage in, garbage out has been used quite a couple of times along, in the conference so far, and I think that's kind of true for us. Whenever we've tried to cut corners on the biochemistry, it doesn't seem to end well. On the right, and then, so pretty much by the time it hits me, we're going to say, okay, the biochemistry, we've got that fairly sorted. So we're going to, the first thing that I will do when I'm given a sample is negatively stain it, and just to have a look. This applies whether it's an 80 kilodalton protein, or whether it's a virus, or whether it's a big fibril, or whether it's anything. Just whack it on a negative stain grid. It takes you 2 seconds, and it will quickly tell you probably whether your cryo is going to be mildly tractable or not. So once it's passed that initial test, then the fun really kind of begins. And I kind of wanted to give you a flavour here of some of the variables and things that we consider when developing a project. So first of all, obviously, we're going to be thinking about the sample. In terms of our buffer compositions, we want to be making sure there's nothing which is going to cause us a large number of problems, such as a high quantity of detergents, lots of glycerol. I have a lot of people that say, I've got 50% glycerol in my sample, is that going to be okay? No, I'm really sorry, it's not. You're going to have to do some dialysis, a lot of dialysis. Sample concentration is obviously a really key one for us. People say, what concentration should I bring it to you in? And I say, as high as you got. And it's easier to dilute than it is to concentrate. So degradation is sometimes a problem, and this is where negative stain can be quite a good initial tool to assess if that's going to be a major problem. And of course then sample heterogeneity. And when I say heterogeneity, I'm talking about both compositional and conformational heterogeneity. So those are kind of some of the things that we will initially look at when it comes to assessing a sample suitability for cryo. So, okay, I've got my protein, I'm going to make grids. What's the next decision? So the next kind of thing to look at is what kind of electron microscopy grid that you use. And I know that there's a huge number of available out there, and I get a lot of emails saying, what should I buy? And for me, the answer is pretty simple. We basically use quantifoil grids. We use quantifoil grids with a 400 or 300 mesh for single particle work. With a hole of 1.2 microns or 2 microns, depending on the sample. We find smaller holes work particularly well with proteins, with anything with a bit of detergent in the sample, for example. We basically use copper grids unless we're doing cell work, and we will consider using support films where it looks like our particle distribution demands it. So, for example, we've had a lot of success in using thin carbon films to get over viruses sticking on the carbon, which some of them like to do. So you picked your grids, you're now going to try and you're going to freeze your grid down. So when you're making your grids, there's a number of different things to consider. I get a lot of emails, so we're very fortunate at Leeds to have both a Vitrobot and a Leica freezing device, and I get a lot of questions about which one do you prefer, which one should I buy, and I just want to talk to you briefly. Well, for those of you who haven't seen both of them, I've just got a quick video of each of them. So as you can see, the Vitrobot is manufactured by FEI. It's probably the most common. It's a double-sided blotter. It's probably used by about a third of our users, something like that. It's fairly, it produces quite nice results. The worst thing about it is the humidifier in our hands, but we just tend to use. Where's out? Wow. Yeah.
Okay.
I like to cause a scene.
So, yeah.
I just thought I'd give everyone a nice little walkabout.
Could you go back to the beginning now, because we've all forgotten what you said?
Yeah, exactly. Yeah, I'll go back to the beginning.
Wasn't it really good outside? You know, the spring.
Sunny, yeah. Apparently it was sleeting this morning.
That's why I arranged the fire alarm, so that everyone could get a bit of vitamin D. Yeah, exactly. I thought, mid-afternoon session, everyone wants to walk around, give you guys a bit of a leg stretch and back in.
Yeah, thank you.
Yeah. Fab. So I hope you all enjoyed that organised fire alarm where I just thought, afternoon session, you want a little bit of a leg stretch, get yourselves outside. Yes, where was I? Freezing devices. So Vitrobot, we've got the Vitrobot, it's a double-sided blotter, it works. We also have a Leica, which is a single-sided blotter, which you can see here in operation. Like I say, pretty much both models, in my opinion, have got pros and cons, but our users are fairly evenly split between the two. Personally, I have a very slight preference for the Leica. I think the way that it's set up gives me very slightly cleaner grids, and I find it slightly easier to use, and I also like the fact I have control over the ethane temperature, but I get very nice results with both instruments, and the only reason I would recommend buying one over the other is if you do cell work, then the Leica, with its single-sided blotting, seems to work better in my hands. But basically, they both work really well, and I wouldn't worry about it too much, basically. So back onto the optimizing the sample prep. I know earlier, basically we use liquid ethane in order to freeze our specimens. We did try ethane propene mixes and we didn't find it made a massive difference, so that's what we've stuck with. But I know different sites do different things on that front. And then once you've got your grids, your protein, you've frozen it, you've got your grids, hopefully you've got a lovely, nice thin ice layer containing your particle. You've now got to screen the thing. So at the moment we can screen in cryo on our T12 or F20. Obviously the T12 we would literally just be looking to say, is there ice? We don't tend to do that too often. And we are actually doing a reasonable amount of screening in our cryos at the moment, especially for the very, very small, very, very challenging specimens where it's difficult to screen on any other instrument. Although our F20 is currently undergoing a detector upgrade and we'll be getting a CETA on that one, and we're hoping that will upgrade the F20's ability to be able to screen different samples. So, imaging conditions. This is a visual representation of my face when I think about all of the variables that you can possibly change when you do your data collection, and it can be a little bit overwhelming. So luckily, mostly when you're Dan or Corey, or in Leeds' case, me, and as of next week, Emma Hesketh, who's going to be joining me in doing supporting users on the Titan cryoses. I just wanted to give you a quick brief run through of the kinds of parameters that we would always consider when doing a single particle project, and what we use as a lead standard recipe, because there may be some differences with other sites. So the first one, which detector? So obviously the two main detectors are your Falcon 2, Falcon 3, or K2, although obviously there's direct electron detectors as well. For us, pretty much, if your protein is above about 250 kilodaltons, we will put you in the direction of the Falcon 3 in the first instance. If it's smaller than that, or it's a tomography specimen, we'll point you in the direction of the K2. However, I think that the lower size limit for putting something onto the Falcon 3 is probably going to go down as we work with the Falcon 3 more encountered mode. So this is a boundary which is a little bit flexible. But they're both very nice detectors that have produced very nice results, and I'll talk a little bit more about the Falcon 3 later. So dose per frame. We mostly use between 1 and 2 electrons per frame. If we want to get to higher resolution or it's bigger, we'll go towards 1. If it's smaller, we'll go towards 2. Total dose. So this is again where we differ a little bit, especially if we're using the Falcon 3 in integrating mode. We tend to work with reasonably high electron doses, and it's not uncommon for us to work 60, 70, 80, 90 electrons per angstrom squared over the exposure. This is a very high dose, but our logic is, well, we fractionate, so you're not going to be losing anything in terms of in your final reconstruction, you can get rid of most of that dose, but fundamentally we're a bit lazy, and it makes auto-picking and doing the initial classifications just so much easier. So we tend to work at pretty high doses. How many images will I get? So there's this bit of a, you've obviously got, so in our case, we've got 2 different detectors. Each of those detectors have got 2 different modes, and depending on how your imaging is set up, they're going to be running at very different speeds. So taking the Falcon 3 as an example, the Falcon 3 in counting mode for most of our standard operating conditions will get you between 25 and 30 images an hour, whereas our Falcon 3 in integrating mode will get you between 100 and 130. So there's a big difference in the quantity of micrographs that you're going to be getting per hour. And then, of course, possibly in the number of particles that you're going to be getting. If you've got an extremely heterogeneous system and you've only got a 24-hour session, then this might then which mode you collect in might not just be determined by what resolution you want to achieve, but also by it's not just going to be about the resolution, but also about the overall particle number. Sometimes you just need quantity in order to be able to do the classification for some systems. Magnification, both microscopes, most of the time for single particle projects we use magnification, which gives us around 1 angstrom per pixel. And acquisitions per hole. So, like I said earlier, we regularly use 1.2 micron holes. We generally do 3 acquisitions around this hole, clipping a little bit of the carbon in each, or standard operating positions would be conditions would be 5 images around a 2 micron hole. So we've put a reasonable amount of effort into developing standard operating procedures for each of our cryoses. We are aiming to, for most people who collect data at Leeds, to be collecting data in a very similar way. Our main logic behind this is that it's if there is going to be, if a problem arises and we're not quite reaching the resolution that we should be, we want to be able to pick up problems as early as possible, and if everybody's doing something in a very similar manner, then it helps. It also means that as we build up our team and as we're having more users from externally and from industry, then we're going to be able to, no matter who is supporting your session, everyone's going to be working to the same hymn sheet as such. So if anyone wants to have a chat to me about this, please come and have a chat to me afterwards. So you've got your data, processing pipeline. I just put this in at the last minute because I realised that not everybody does things exactly the same as us, but basically when we've got the data, everyone for single particle projects pops it through GCTF, Motion Core 2, and then does all the processing in Roland 2. And we're currently working very hard at trying to implement an on-the-fly processing regime, which is going to be hopefully up and running in the short to medium term. Fab. So assessing data quality. So you get your data off the microscope, and sometimes sometimes you don't necessarily know whether there's going to be a problem until right at the end, and you're like, why has my map only got to 5 angstroms? You know, what was going on? So so a number of people in different labs have been looking at developing tools to assess data quality as it comes off the microscope. So Matt Yadans has been particularly leading on this front, and has developed a script which is available on GitHub for assessing micrograph quality. So behind me we have 2 examples, one of a bad data set and one of a better data set, but it's still not very good. So this basically the top plot shows you your defocus, and then along 2 axes, and then this is going to be telling you, the spread of this is going to be telling you about your astigmatism, and we can see here the number of, this is a histogram of your micrographs and what the astigmatism is. So we can see on this one was really badly astigmatic, and you can see when GCTF kicks you out what it thinks the resolution in those micrographs extends to. The histogram is not tightly packed around the 2 to 4 angstrom mark, which is what we would normally like to see, but it extends out hugely. And what was actually going on during this data set was that our water cooling system was blocked, so the lenses weren't being appropriately cooled. So we can see here what a massive impact cooling the lenses has on the performance of the microscope. So we can see here on the right that we have a better data set. However, this second hump here in the resolution, it probably does indicate that something was behaving suboptimally. And if we have a look at this final one, this final plot, we can see that this would be much more what we'd be shooting for. So we've got a nice tight distribution in terms of what resolution GCTF thinks the micrographs extend out to, and centered here just below 3 angstroms. So you notice this plot is very slightly different to the ones on the previous page. So this is Matt's updated script, which is not available on GitHub, but which has which basically also plots you some statistics over time. So if anybody wants to go and have a chat to Matt about that, I think he's very happy to talk about it. Another tool which Matt has developed, and the first script is available on GitHub. A second tool which Matt has developed is a really interesting tool for finding crystalline ice in images. So no matter how diligent you are with trying to remove the holes at the edges of squares, you will always get some images which have got crystalline ice in. So Matt's developed a tool which basically takes the micrograph and takes the power spectrum and generates a radially average power spectrum, and then fits a line to this, and then looks for any signal which extends, which basically deviates from this line at the spatial frequencies where you would expect to see a signal for crystalline ice. So here, for example, is a nice clean micrograph, and at the bottom here, this extra hump is telling you that you've got a serious crystalline ice problem in your micrograph. And again, it can do clever things like give you this over time. And this might be a really handy tool for you to basically get rid of these micrographs before they even go into your data processing pipeline. And this is something that we're going to be looking at implementing in our on-the-fly scripting. So I just want to finish off with some thoughts about the Falcon 3, and then we can go back outside into the sunshine. No fire alarm needed. Talking about the Falcon 3. So most people who've got the Falcon 3, including us when we first got it, have been extremely excited about using the detector in counting mode. But I'd just like to share with you a couple of examples which are leading us to think that counting mode might not always be the best way forward. So Sean in Steve Munch's lab collected 2 data sets on yeast IGPD, as I spoke about earlier. He collected one integrating data set and one counting data set. And you notice that these, in terms of time, were basically exactly the same, but the difference in the number of micrographs was quite significant. So Sean processed this, and the take-home message was that the final resolution, so I'm going to caveat this and say that the resolution of this structure is almost certainly limited by preferred orientations. It's not an ideal test specimen, and I'm not saying that integrated will get you to where counted does, because undoubtedly counting behaves better. But for this particular biological specimen, a similar time collection on both, you ended up getting to a very similar answer, although the integrating mode just edged it. And if we have a look at the micrograph analysis for both of these, we can see that the performance of the microscope was stable for both, and in terms of the local resolution, the maps look very similar. The final example I'd like to share with you for the Falcon 3 is a structure which Emma did so Emma put the sample in the microscope on a Wednesday and had the 2.7 angstrom structure the following Wednesday. So this went from going into the microscope to having the structure, well, having the map within the week, which was really great for us. Yes, it was a 48-hour data collection. Collected the micrographs, processed them in the normal way, just did one round of 2D to get rid of anything which AutoPick had accidentally picked, worked through the whole stack through AutoRefine, and it just dropped out at 2.7. So I think at some point Emma's hopefully going to be going back to the data and seeing if we can push this a little bit lower. But the fact that this fell out so nicely at 2.7 angstroms on the integrating mode detector was for us really great, and it showed to us that the Falcon 3 almost certainly works very nicely in both modes, and it might not necessarily be that counting is always the best way forward, especially for a system where you might have a high level of heterogeneity, and in a given session you want to collect enough particles to be able to classify out your thing of interest. So I just want to finish off by saying that access to the facilities at Leeds is available. We offer up to 20% of our time as a service to external users and to industry. So if you'd like to know more about that, please come and find me. But I'm just going to finish off by talking a little bit about our booking system, because I know that this is something a number of people are looking at the moment. So the booking system that we use is ARIA, which is by InstructHub, and basically all of our internal users use this to book all of our electron microscopes. But as an external user, you can go onto this website, you can fill out a form with some very, very basic information. All we're basically looking for is to check that there's no conflicts of interest with any of the projects that are going on at Leeds, then basically one of us will be in touch. And it's quite a nice system, and it works very well for us, and it's got nice reporting functionality. So if anyone is looking at changing the booking system within their facility, this is an option worth considering. With that, I'd just like to finish and thank all of the wonderful people who have done all of the maps that I've shown today. So obviously I'd like to say a big thank you to my boss, Neil Ranson, who is the Director of EM at Leeds. And in Neil's group, we have Emma, who did the beautiful CPMB work, the Gemini work, and the lutivirus work. Matthew Danza, who's our scripting man, amongst also working on some really exciting membrane protein projects and amyloid projects. Dan Hurdis, who is also in Neil's group and did the lovely work on the BK polyomavirus. In Steve Munch's group, Sean and Rachel have presented work from both of them there. And this is my favourite photo of all time, just ever. As well at Leeds, we also have Hon Fontana, who I haven't spoken about his work today, but he's doing some really interesting stuff with electron tomography that we're super excited to get going with as soon as Cryos2 wakes up. Finally, I'd just like to say a big thank you to the guys at FEI. So I know that we've certainly had a lot of problems, but fundamentally their engineers work super hard and have been, on a personal level, amazing. And a big thank you also to Sarah, who came and did our initial app specialist training. Yeah, that's everything. Thank you very much.